Hello and welcome to Think Supersystemic. We explore the science, art, and economy of interconnectedness. Today our guest is 14-year-old Nina Khera, longevity researcher and entrepreneur. Yes, you heard that right, 14 years old. She comes to us from the Knowledge Society, which is a group you'll learn about in just a moment. Hi Nina, welcome to Think Supersystemic.
Hi, thank you for having me.
Thank you so much for joining. So I wanted to talk to you. About generally first, how did you get started in what you're doing? I mean, you're 14 years old, which you're obviously doing things that most 14-year-olds don't do. Can you talk a little bit about what kind of brought you into this world of like exploring science and exploring, you're really exploring your full potential at such a young age?
Yeah, so from a young age, I've always been really interested in coding and generally computer science, just because, I was really exposed to that as a child. And it was something that, really fascinated me, just how computers work, what can I find out about the world using these sort of technologies. And yeah, when about 2 years ago, I joined a program called TKS and the Knowledge Society. It's sort of an incubator, sort of a human accelerator in a way for kids aged 13 to 18. And pretty much I joined, and a topic that we learned about a bunch of different topics, and a topic that I found really fascinating was human longevity and aging. And because of both TKS and like just learning about that technology and general area of science, and also experiences that I had with diseases that were related to aging years prior. And so kind of from there, I did a lot of different research into it. I read a lot of papers, and I ended up kind of coming up with an idea to go deeper into this field in March, the next year. And yeah, and then from there, I guess I've just been kind of continuously learning and about this field.
Awesome. Can you talk a little bit about TKS, kind of like what, when you joined up with them, what types of events do they do? Like, how do they obviously it sounds like it's a sort of like a support and growth group for people in your age group. How does that work? Like, what do they do? And like, how do you engage with other people? And have you met lots of people like yourself? Because I imagine that's quite difficult in your local areas and stuff.
Yeah, so TKS pretty much, they're a sort of human accelerator who teach kids about different topics, so different topics within areas of science, technology, and also not just about those topics, but how to do something with those topics. So learning about how to do real projects and how to work in teams, a lot of soft skills, presenting, creating one-pagers, decks. So we've, as part of TKS, we get a lot of experience with, consulting work. I've consulted for Walmart, Wealthsimple, Kidogo, an organization in Africa, and Wealthsimple is a banking organization, and Walmart, I'm sure you know what Walmart is, but a chain of general stores. Yeah, so TKS it's a great program because it's really easy to meet people who are really motivated there, because the application and kind of the actual entering the program does require effort and that kind of stuff. So that the people who come in are really dedicated, and people who stay in and who are really excited about their the work they do. You can call with them. They're called brain dates, and basically you just talk to people for 30 minutes to an hour and learn a lot about them and anything you want to, really. And yeah, I made a lot of really great friends there, and it's a really amazing community. That's one of the key appeals for me too. I get to meet a lot of people who are my age and working on things that I'm really interested in too.
Awesome.
Yeah.
Very cool. And so can you talk a little bit about, so you mentioned that you did some consulting work with Wealthsimple, Walmart, and this organization in Africa. Were those great experiences? Did you learn a lot? Like what types of things did you learn from those experiences? It's really interesting to have. Yeah, good.
Yeah, so pretty much in terms of these consulting experiences, I've learned a lot because you typically form teams, and I've learned about how to work in a team and kind of get something done before a deadline really well, high standards. And yeah, a lot of the time it was really cool because it was just really a problem-solving experience, right? Because basically they just kind of posed a problem to us. They said, how can we maybe increase our profits or it really depended on the organization. Sometimes people were like, how do we cut our expenses, that kind of stuff. And, they gave us a lot of data to work with. They gave us sort of a data room where we sort of went in and we came up with ideas, and a lot of people came up with really, really great ideas that are actually being implemented, which is really exciting. And Yeah, I mean, it was a really, really good experience because I learned, how do I strategically problem solve? How do I use the data that I have to come to a really good conclusion? And also, I just learned a lot about, working with teams and also, just kind of making Jackson a lot of soft skills.
Very cool. So yeah, now I'd like to transition to talking about the project that we specifically worked on, like the microbes versus dementia, and maybe there's a connection there, that sort of thing. Can you talk a little bit about what you found when we asked you to kind of take a closer look at that stuff, and what was surprising, what wasn't, and that sort of thing?
Yeah, so just do you mind if I quickly explain like the project and that kind of stuff?
Yeah, please
Um,
do. Yeah.
Yeah, so pretty much we were looking at different concentrations of different bacteria in the gut microbiome, but just the general microbiome within these different aging-related diseases. So I was looking at it within dementia, specific types as well. Parkinson's, Huntington's I looked at as well. There's a lot of interesting diseases that I looked at. Also cardiovascular diseases because those are really related to aging. And then from there, I kind of collected a bunch of data from studies. So I compiled a bunch of data on, so how, I guess almost how many times, so I guess we'll go first off, what bacteria was found generally to be kind of in sort of a dysbiosis sort of state. So what bacteria had too little of it and what bacteria had too much, which bacteria should generally avoid. And I sort of compiled it in a spreadsheet. I plotted the results and basically used kind of a +1, -1 sort of system for the quantities of bacteria. But yeah, so I ended up finding really, really strong connections between the lactobacillus species and dementias, and generally dementias, but also just general aging-related diseases that seem to be mentioned a lot. And there were a lot of really interesting bacteria that I found had higher higher kind of rates and higher prevalence in these diseases.
Mm.
And yeah, it was a really interesting experiment and just general kind of data collection experience to do, and I really enjoyed it.
Very cool. So was it sort of like there were certain species that had almost become pathogenic in people, and certain people with that would eventually end up with dementia and that sort of thing?
So I analyzed a lot of studies where they looked at the risk for dementia as well. So what is the risk for dementia and what is the risk for these diseases in relation to the actual and in relation to bacteria? And so there it is shown that many different bacteria, like the balance and the biosis of them. So there's a lot of different states of dysbiosis that actually occur in these diseases. Sometimes they occur after the disease has started, which I find to be a little less useful, but it could also mean that it furthers the prognosis and furthers the progression of these diseases. And then also there is, before the disease actually happens, like they could be causal factors, which I think is really cool, and I want to further investigate that.
Oh, very cool. Can you talk a little bit about dysbiosis of the microbiome versus a healthy microbiome? Like if you were to take a sample of somebody's microbiome and or two samples, one person who is healthy and one person who has dysbiosis of some sort. Can you talk a little bit about what that looks like? Like how is it a matter of the number of species, or is one species taking over or something like that in the unhealthy person?
Yeah, so in terms of dysbiosis and in terms of the microbiome in general, so typically there's a healthy balance to have all your microbes at, and that is sort of found when people do microbiome tests. There's a lot of them out there actually. I think Biome's one. There's a few more, and pretty much that does kind of maybe kind of increase the likelihood that you might get a certain disease if you have different dysbiosis. And different bacteria kind of are fed and by different fibers. So it's also kind of thinking about how do you feed your microbiome too, because there's that sort of there's this sort of balance between the balance of the microbiome that you kind of want to hit. And generally it's about what you feed your bacteria, of course, because really a lot of your diet choices and your food preferences are actually kind of decided by your microbiome, which I find really awesome. But yeah, so pretty much in general with the microbiome, the thing about dysbiosis is that typically when there's different bacteria do different things to your body, right? So when there's too much of a certain bacteria, I mean, in general, everything's good with moderation, but when there's too much of a certain bacteria, too much of a certain phenotype starts happening. And that it's not really the bacteria itself that caused the disease, but it's more the bacteria cause, phenotypes, and they cause different things down the road that cause the disease ultimately. So yeah, I wouldn't say it's like immediately, this bacteria, because this bacteria is building up, Alzheimer's immediately happens, but it's more, as a result of, many years of this happening, Alzheimer's is, way more likely to occur in relation to other, senescent cell count and different other phenotypes of aging that are happening.
Okay. I guess that speaks to the complexity of the human body and that sort of thing. Have you, since you've learned a lot about the microbiome and stuff, has it influenced the type of foods that you eat? I mean, are you telling your parents that you want to eat more gut-friendly foods and that sort of thing?
I do try to eat more fiber on a daily basis just because it is really good for your bacteria. Yeah, I mean, it sort of has. I wouldn't say like, I make every food decision. I like to, relax once in a while. I don't make every food decision based off my bacteria, but for sure I do, try to incorporate fiber friendly, foods with high amounts of fiber into my diet, which is definitely helpful.
Wonderful. So you mentioned briefly there, you mentioned senescent cells, and I understand that you want to kind of go deeper into researching senescent cells and so forth. Can you give a brief overview of what a senescent cell is? Because it seems like it's something that's really important to aging and stuff.
For sure. So senescent cells are sort of zombie cells in your body. So basically what happens is when your cells divide, they copy DNA from chromosome to chromosome. So kind of like a little strand of DNA. And so when they copy this DNA, there's this sort of error within DNA replication. And pretty much at the ends of your chromosomes, a little bit of DNA gets cut off. And, as you may know, DNA is sort of very valuable for you because it basically does not decide your genetic information. It decides who you are as a person. So losing that is definitely not a great thing. And so, so your body has to obviously develop some sort of defense mechanism for this. Otherwise, we'd all have random mutations. What your body's developed is actually these things called telomeres. And telomeres are sort of like eyelets on the end of your shoelaces. So those little plastic bits.
Mm-hmm.
And with every cell division, they get cut off a little bit more and more and more until they run out. And that's after about 50 to 70 cell divisions, they hit the limit. And pretty much what happens then is your cells go into a state called senescence. And this is replicative senescence. So basically this entire process I just described with them dividing over and over and over and the telomere shortening is something called epigenetic senescence. So this can have an effect on your genes, has an effect obviously on your cells. And that's what it's the process of them getting closer to senescence. And that overall, senescence is replicative senescence. And that's where like your cell just shuts down, does not divide anymore. And that's when it starts releasing this thing called SASP. So senescence associated secretory phenotype. And pretty much what this does is it actually, it's a bunch of proteins, it's a bunch of inflammatory cytokines, chemokines, a bunch of different little molecules that can A, promote inflammation, which is not good for aging. B is also, it also linked to cancer because senescence is almost the opposite of cancer, if you kind of get what I mean, because senescence is sort of this proliferative nonstop, but cancer is sort of proliferative nonstop, and senescence sort of just shuts down the cell, right? So senescence also has molecules that it releases that kind of can almost encourage cancer in other cells. It's definitely not a great cell state to be in. While it does have some pros though, it's not totally negative because it does promote faster wound healing. And it also does, cause, it can prevent cancer in the cell that it's induced in, because obviously if a cell is senescent, it's not going to immediately becomes cancerous because it's sort of shut down. I mean, I guess it's sort of a trade-off, but it's definitely, of course, better than the cancer.
Got it. And so as we age, we get these senescent cells proliferate through our bodies and we become more inflamed and so forth. And that's part of the aging process, right?
Yeah, a huge part.
And is it, so the challenge, am I getting this right, that the challenge is that we want to learn how to get rid of some senescent cells, but not all of them or a certain number of them and that sort of thing?
Yeah, so for sure we shouldn't get rid of all of our senescent cells. A, because wound healing, the benefits they bring. Also, sometimes senescent cells kind of gather around certain organs. They actually kind of can be protective in some cases. So they're not all bad. Of course, nothing is all bad. But the thing about senescence is that we do want to get rid of a lot of our senescent cells because they can cause this inflammation, the SASP, the SASP that they kind of release. is definitely not a good thing for our body and can, contribute to a bunch of diseases in many different ways, actually, because, it can actually contribute to, plaque buildup in cardiovascular diseases. It can contribute to, our neurons becoming well, not our neurons can't become senescent per se, but the glial cells and the supporting cells that kind of support our neurons can become senescent, and that is really bad for diseases like dementia and Alzheimer's. So they cause a lot of different things. I feel like everything in aging that we're investigating right now, all the hallmarks actually cause different things that actually lead to this, these bad these diseases. So it's just it's really interesting in that sense. But yeah, we're trying to get rid of most of our senescent cells in almost the right places.
Great. So would you say that longevity research and so forth, it sounds like, from what I understand of some longevity researchers that I follow and so forth, is that they're trying to get longevity or to aging treated as a single disease in itself because it has all these other kind of downstream diseases that come from it. Would you say, how far along in that process would you say we are? I mean, are we 15, 20, 50 years away from really understanding aging and slowing it down to such a degree that people live much healthier to, a longer degree?
Yeah, so I think that's a really interesting question because the thing right now about aging is that we have a bunch of different theories, but we can't agree on one. So basically, there's a theory about epigenetics. So that's a theory of information loss. So pretty much, there's this professor. I'm going to give a few examples. This is a professor at Harvard, David Sinclair, and basically what he's kind of thinking of is that basically due to epigenetics, that thing I mentioned earlier. So basically, that's your gene expression in cells and how it changes as you age and just changes in general due to chemical tags. And pretty much, basically, he kind of stipulates that basically it kind of your epigenetic tags and your chemical tags kind of change as you age, and that is what leads to the actual phenotypes you feel within aging. Then there's some other ones that kind of say that there's combined theories. So there's genetics. So basically it's your genes, what you already have. It's your entropy, so that's sort of physics. Basically the amount of time that you have. The amount generally of because entropy, the second law of thermodynamics, states that everything, the entropy will only increase. So entropy is sort of the amount of a system that's almost kind of unproductive in a way. So basically that's sort of the general law of physics that people kind of link with aging. And then there's last of all lifestyle. So, how do you live and how does that kind of influence what goes on in your body? And I think this, obviously the third one is the most controllable, but we are kind of figuring out ways to control the first. So, I mean, there's a lot of different, theories of aging at the moment, but they're all kind of really interesting. I'm really excited to see what happens, but that does mean we're really far out from, once we actually pinpoint a theory, then it's about, bringing that theory to the World Health Organization and saying this is what aging is. And, like, how do we get to the point where you can classify aging as a disease? Because unfortunately, that's not the outlook we have right now. And also, it's also about, I think the way to kind of find a good theory is that you have to do a lot of research. You have to do a lot of kind of understanding of go from kind of first principles. Like what are the most basic experiments that we could do that would, tell us a lot about aging? Because right now, there's a lot of really, really complicated experiments being done, and it's sort of extending on to current theories. And the way I see it, theories are a bunch of trees, and we have a lot of trees in this aging forest at the moment. But once we kind of somehow consolidate all of these into a single really large tree that's sort of with a lot of branches that we really, really understand. We bring that to the World Health Organization, we bring that to the FDA. Because right now what we're actually doing with aging trials is we're trying to pretty much kind of go look at diseases related to aging and basically create trials based off of that, but that's not really productive for aging in general. I mean, it is in a sense, but it's also just, I wish we could test it on actual aging. Then again, 70 years might not be the ideal study time, but it still would be really interesting, right? So kind of being able to classify aging as a disease to the FDA and WHO.
Very cool. And so there are a number of aging clocks now, right? People are trying to develop these ways. Can you talk a little bit about aging clocks and like where we are with that?
For sure. So right now, in terms of aging clocks, there's a lot of really interesting ones out there. So I know the original one was created by Dr. Steve Horvath at UCLA, and pretty much that was around epigenetics and DNA methylation. So basically that is, a key component of epigenetics and epigenetic change. And it was really great. And right now we're getting a lot of improvements in both in accuracy because, his clock it was probably a 5 to 10 year difference from the actual age. So it was actually really good. And we're actually making a lot of improvements. I've got to hear about a lot of different clocks that researchers are creating, and it's been so cool to read about these and also just to kind of hear about these in general. Yeah, I mean, I think we're kind of getting to the point where we could potentially, put an epigenetic clock out there. Another thing that's really interesting is bioage tests, which are very similar in a sense.
Mm-hmm.
And those are basically, there's one that's epigenetic out there, I think. It's Index by Elysium. That one's really interesting. Basically, they're looking at, your epigenetic markers, and they're basically kind of creating what is your biological age based off of these, which is so fascinating.
Mm-hmm.
So, I mean, I don't know if I would call that a clock per se, because aging clocks, the purpose could either be, to find your biological age, but also to kind of find your chronological age based off of your epigenetics, right?
Mm-hmm.
So it just depends on the purpose, I guess. But yeah, bioage tests are really cool too.
Cool. Would you say it's, would I be correct in saying that these ways of measuring biological age is essentially measuring your current kind of risk profile as it relates to your lifespan? Is that essentially what it is?
Yeah, I'd say so. So pretty much, I guess in terms of risk profile, it's really about, yeah, I'd say so. So pretty much the thing about biological age is just, what people are doing is looking at, well, first off, epigenetic markers. So they can make those like that. They can make that numerical data, and based off of that, kind of run some statistical models and kind of come up with their biological age based off of that. There's also, just different concentrations of, there's metabolites. So I know a lot of people are studying metabolic pathways. So yeah, I guess really the kind of the productiveness of biological age is to find out, what is your risk? Like what is your risk profile for these different diseases and that kind of stuff. So yeah, I'd agree.
Cool. Great. This is so fascinating that you know so much about this area, and I'm so excited to see where you go in the coming years. I mean, you have so much ahead of you. Can you, what are your plans for the next couple of years? Like by the time you're 16 or 17, what do you hope to have accomplished? And so for then beyond there, where do you see yourself going?
Yeah, so right now I'm working on something called Biotein, it's just a startup I'm working on, and that's something that I really want to see grow and develop because it's around senescent cells, and it's also we're kind of basically senescent cells are sort of our short-term goal. We're looking at finding biomarkers to measure the concentration of senescent cells in your body at any given point, and then from there we want to create bioage tests. We want to kind of almost also help with clinical validation. So validation for studies that are being conducted in relation to aging-related diseases and dementias, kind of providing more accurate validation than what the clinical analysis and the evaluations that are currently there. Yeah, and that's something I'm really passionate about, but also, we really want to expand to dementias too. So we want to basically find a cure for dementia, which is a very difficult thing to do, but it's definitely something that I'm really passionate about, and I know my team is as well, and we are all really excited about this. So that's something we're doing. Yeah, so that's something I really want to see grow and develop by the time I'm 16 or 17. Yeah, and I also think I want to learn a lot more coding languages too, because I think I want to learn R, and I want to learn a few more languages, because I think right now I know like Python and a few others, but I definitely want to learn more that are relevant to like biology and protein modeling and that kind of stuff. And kind of get into that. But also, in general, I just kind of want to expand my skill set too, because there's so much more I can learn out there. There's definitely a lot more, and I feel like I need to be constantly gaining knowledge and constantly getting understanding, because then I'll have a really up-to-date worldview, and also I just love learning, so I'm really excited.
Awesome. Well, thank you so much for doing that work, and also thank you for doing that little project for us, and thanks for joining the call today to discuss all that you're up to. It's so fascinating, and I really appreciate what you're up to.
Thank you so much.
Okay, thank you. Have a great one.
You too.